the big reason why we still maintain the wild wheats, well, other than it's very hard to get in a lot of areas to collect anymore. For example, I tried to get some seed from Iran a couple months ago. I had a gentleman from there that wanted to trade seeds with me, and I said I'd love to. We don't have very many seeds from Iran, and the U.S. government won't even allow me to bring seed in from that country. Secondary reason is wheat breeders have been breeding between wheat cultivars for years to make new cultivars. So let's say a cultivar from Kansas doesn't have leaf rust resistance, but a cultivar from Texas does, so they'll combine the two and they'll make a new cultivar that has resistance to that. Well, wheat breeders have been doing this for 100 years now, and what we're finding is what we call a bottleneck, in that we're kind of using up all the genes that are in wheat, that are in cultivated wheat as we know it, in the wheat cultivars. And so where do we go for new ones? Well, the answer are these wild species. And they've been growing separate from wheat, so they may have genes that are not found in wheat. And so, but we don't know that. And the only way we know that is to go through the collection, screen them for the disease we're interested in, and say, aha, there's good genes in that plant that are effective against the pathogen or the bug that we're looking for. And so that's the way we bring new genes into the wheat population. And we've done this. Leaf rust is an important disease in Kansas. Probably one of the, when I came here 30 years ago, probably the most important disease. And we've brought many genes from these wild species into wheat to help combat the leaf rust fungus. We in the lab need to cross the wild species with wheat. That hybrid is never fertile in itself, and it's never viable for much more than 14 days, so you have to extract the embryo out and grow it on artificial media. But once you have that plant growing, you can back cross it to wheat for a couple generations, and you're almost 100% wheat again. So that's our job, about 2-3 years to get a gene in a background that a breeder wants to use. Then the breeder takes that. The breeder doesn't like probably the genetic stock we made in addition to the leaf rust resistance, because we are just combining two plants. We don't know what else is in there. They're not good quality wheats. They're not the best performing wheats probably, but they have the gene that the breeder wants. So he wants to put that in his elite variety. And so he'll then take that for another few years until he gets it into one of his elite varieties that he's ready to release. So while I want to say some of these, I started creating these genes 20 years ago, some of them are still being used from those stocks and still being put into wheat cultivars. We know that the hybrid is usually going to be non-viable. That's an easy step. We've been dissecting out embryos and growing them on artificial medium for a long time. So that's really not the problem anymore. The problem is, A, finding the disease or pest resistance you're looking for in the wild species. So leaf rust resistance, no problem. There's a little fly called Hessian fly. No problem. We found lots of resistance genes to those. Start looking at virus diseases. Really hard to find resistance in some of these grasses to virus resistance. I don't know why. Maybe they weren't exposed to the pathogen like they are now. So that's one problem. And that's probably the biggest problem. And also, the next problem would be, once you cross the wheat, you have to retest it all the time to make sure that you're following the gene along in the population, because you're getting chromosomes from one plant and chromosomes from another plant, you're combining them. Well, you may not always get the gene transferred to the next generation, so you also have to follow the gene you're looking for in every generation. Now, back in the olden days, now, I had to, when I made a hybrid, I had to subject it to the leaf rust pathogen again, make sure it was resistant, only cross those plants that were resistant, get seed, regrow those, inoculate them again with leaf rust, do the same thing over and over again so I could make sure the gene was following me all the time. Now, we can cut a leaf off, do a DNA analysis and not have and just be able to select from thousands of plants, where our gene is and which plants to keep.